Okay, so I think we can make a start. So good afternoon, everyone, and welcome to Paleoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year, generally on the last Thursday of each month, unless they overlap with the holidays. If you're interested in presenting a new method or software you have been developing or implementing, please feel free to reach out. So before moving on, just a few quick introductions. My name is Lewis Jones, and with me today I have Erin Dillon, Will Gertie, and Bethany Allen. The four of us are on the organisational team of Paleoverse, an organisation aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about the Paleoverse, please check out our website and GitHub organisation. I believe those links are just being popped into the chat now. But that's all from me for today. Erin will now give you some helpful tips and instructions for this webinar, and then will introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. So thanks so much, Lewis. As Lewis mentioned, the format of this seminar will consist of a talk by our speaker, followed about 10 to 15 minutes at the end that will be dedicated to Q&A and discussion. During this time, my fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and that it will be made available on the Paleoverse YouTube channel. However, we will stop recording prior to the Q&A period in case you prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You'll find a Q&A button. You can use this to submit questions to our speaker, and during the Q&A session, we'll either read out your question or invite you to turn on your microphone if you'd like to ask your question directly. There's also a raise hand button. So alternatively, you can raise your hand and we'll call on you and invite you to turn on your microphone so you can ask your question. There's a chat function. So currently you can only chat with the host in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And then finally, there's a closed caption button. So you can turn that on to view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Paleoverse Code of Conduct, which can be found on our website, and Bethany is now putting a link in the chat. So with that, I will pass it over to Will to introduce today's speaker. Over to you.
Thanks, Lewis and Erin, for getting us all up to speed on how the lecture series works. Today, I have the truly great honor of introducing our speaker, Dr. Richard Stockey, a lecturer in paleobiology at the University of Southampton. Rich's research primarily focuses on using numerical and statistical modeling approaches to better understand how environmental change has impacted marine ecosystems through Earth history. Much of his published research has focused on how Earth's oceans became habitable for marine animals over the last billion years. More recently, though, he has also begun working to improve our understanding of the similarities and differences between more recent ancient warming events, such as those of the last about 300 million years, and modern anthropogenic climate change. In addition to his research, Rich is also actively involved in developing software and tools to facilitate paleoclimate modeling and analyzing the outputs of such modeling, some of which I believe we'll hear about today. So without further ado, we're really excited to hear from Rich today with a talk titled Modeling Ancient Animal Habitats in Space and Time, Linking Fossils and Earth System Models. Please join me in welcoming Dr. Stockey to our virtual stage.
Awesome. Thanks, Will. So hang on, let me just share my screen, which I've now lost. I didn't expect everyone to disappear. That's great. Okay, so hopefully you can now see my screen. Lewis, can you give me a thumbs up?
Looks great.
Great. Okay, so firstly, I'd like to thank all these people and probably many more who could be listed here. I'm going to talk about a few different mainly in prep studies today and in prep nearly finished software package. But you know, it takes a whole village. So not all of this is just me. In terms of an introduction to me, I'm Richard. As Will said, I'm a lecturer in paleobiology at Southampton, where I started 2.5 years ago. I use he, him pronouns. Before that, I was at Stanford University, where I did my PhD around the same time Will did. And my research really sort of focuses on these two key questions of how has environmental change impacted marine animal ecosystems through geologic time, and how can we best use the fossil record to understand threats of anthropogenic climate change for modern ecosystems. So I wanted to give a little bit of background, as I often do, about my research and why I think it's worthwhile or important and what motivates me. So I'm really interested in trying to understand how environmental change, particularly in the oceans, impacts biodiversity. Again, particularly in the oceans, but lots of these problems apply. The problems I'll talk about today apply to terrestrial environments as well. So when we try to study the interaction between environmental change and biodiversity dynamics in modern oceans, that poses a huge number of challenges in terms of the sheer vastness of the data available to us, or the sheer vastness of the possible data available to us, and the limitations on collecting and then analyzing that data. When we think about trying to understand how environmental change has impacted biodiversity in Earth's past, which I think lots of people who are attending this seminar series are hopefully interested in, that poses a number of other challenges. And probably the most obvious one is that we're always dealing with indirect records of the systems that we're really interested in. So in terms of environmental change, we look to the geochemical record. So often that means that we're looking at indirect records of marine environments that are preserved in sampled and exposed sedimentary rocks. When we're thinking about the fossil record, we're interested in ecology and evolution, and we try to reconstruct biodiversity dynamics, again, based on sampled fossil occurrences, which are going to be impacted by a number of different processes. So how can we make these two different indirect records speak to each other? Principally, I would argue that there is always merit in being a good interdisciplinary scientist. So to understand what the geochemical record means for environmental change, we need to educate ourselves in oceanography to be able to link geochemical proxy records to seawater conditions. And then similar principles apply to linking the fossil record to ecology. So we need to educate ourselves in macroecology, physiology, and evolutionary biology in order to translate this indirect preserved record that we see in the fossils into the kinds of processes we're really often trying to ask questions about, certainly that I find myself asking questions about a lot. And then sort of the thing that I get really excited about doing is trying to make these processes interact in a model world. So trying to understand exactly how our fossil record of biodiversity and our geochemical record of environmental change might coexist in a fully functioning system. And that's where I get really excited about using 3D or 4D ocean atmosphere models, where we can allow simulated ecosystems based upon the data we get from the fossil record to interact with reconstructed environments, again, based on the data available to us in the geochemical record. To think about it graphically and in space rather than in words, another big problem, often when we're trying to link ancient patterns of environmental change to ancient patterns in biodiversity, is that geochemical proxies aren't always recording on the same spatial and temporal scales as the fossil record. So for instance, when we're thinking about ocean oxygenation, which I spent a lot of time thinking about, our geochemical proxies recording these processes are generally most sensitive to the area of anoxia and suboxia, so not quite no oxygen, but some, on the shelf slope, so sort of at mid-ocean depths, shallow to mid-ocean depths. In contrast, marine animals, the vast majority of marine animals today, probably the vast majority of marine animals in the past, and certainly the vast majority of our preserved fossil record, is of life in shallow shelf marine environments. So we get this sort of spatial offset in what our geochemistry and our fossil record are recording. If we're able to constrain 3D models, such as the kind of paleoclimate and paleoceanographic models I'll talk about in a minute, using our geochemical data, we can then investigate what that means for the slightly offset environments where our marine ecosystems are actually living. And this process is what I spend a lot of my time thinking about, and what a lot of my research effort and people who work with me's research effort is directed at. So in terms of sort of how we're able to do this and why we're able to do this is really, really reliant on sort of bare bones grassroots geology. We need to know where things such as where continental plates were at different times in Earth history. Our spatial models are always going to be dependent on those kinds of constraints and obviously the uncertainties associated with them. We're now in a position where, as a result of a number of different digital elevation models or topographic models through Earth history, we have a number of different explicit spatial hypotheses about how the positions of the plates have changed through time. We also have lots of different hypotheses about how Earth's climate has changed through time, so how things like atmospheric CO2 has changed through time. What that sets us up to do is to begin providing global circulation models, climate models, paleoclimate models with the kind of information that we would provide modern climate models with. So we need a physical environment, and then we need boundary conditions such as solar flux, greenhouse gas composition, that kind of thing. So with these other lines of evidence coming together from either sort of tectonics and field geology, really, and geochemistry, we're able to start using the kinds of tools that modern climate scientists, ocean dynamicists, atmospheric dynamicists use. And that's kind of the reason that, as I say here, we're only quite recently able to do this kind of research. And a lot of this is facilitated by actually quite a small group of paleoclimate modelers. So a key group would be the Bridge Group at Bristol with Paul Valdez, Dan Lunt, Alex Farnsworth. And they specifically have tended to use a paleo-tuned version of the HADCM3 model, or specifically the HADCM3L model, which is an old version of the UK's CMIP Climate Model into Comparison project model. Coupled model into comparison project model. So HADCM3 is an older version of that Met Office model. And for the first time, they have been able to run spatially resolved simulations of ancient oceans and terrestrial environments based upon plate reconstructions and reconstructions of things like atmospheric gases, solar constants, etc. And that's provided a lot of inspiration to paleontologists and geochemists, me included. And the ability to actually simulate these 3D environments has allowed us critically to start tying point data, so paleobiological data such as fossil occurrences from the paleobiology database or point-based geochemical data, to simulated climatology, so simulated environmental fields. And this is pretty new, right? So of all the papers that I've screenshotted here, there's probably nothing older than 2018. And that makes a lot of sense because actually a lot of these model simulations aren't much older than that. If I were to flag what I think has been really, really important to me, it's this Phanerozoic series that I have a GIF of ticking over on the screen right now. So here what we've got is the first stage by stage series of Phanerozoic climate models. It was produced by Paul Valdez and published in this paper in Climate of the Past. And a very similar model series to this, which was produced by Dan Lunt, was used in this paper, which I think is in many ways a sort of new gold standard for this kind of research, by Cooper Malinowski, Aaron Saup, and others, where they indicate that climate change and global temperature specifically is an important predictor of extinction risk on Phanerozoic timescales. And they did that by tying thousands of fossil occurrences, paleo-rotated, to the climatologies in a model series, very similar to the one ticking over my screen right now. So in terms of where I tend to fit in, because there are lots of people doing brilliant work, these high results HADCM3 models are incredibly cool, and there are lots of people who are probably better quantitative paleobiologists than me, interested in these kinds of problems. So where I try to fit in is kind of in the middle. So really in building techniques to improve our reconstructions of ancient marine environments, but also how we link them to fossil data and how we can test those models, because those models are always experiments, they're hypotheses, using geochemical data. And that's really this central box of the graphic that I have here. So in terms of sort of problems that I like to work on and things that have motivated me, a key thing is that some of these spatially highly resolved models are brilliant for some scientific questions, but they're not a great fit for all scientific questions. So for instance, these spatially well-resolved models are very computationally intensive. They take a long time to run on quite big supercomputers. But in addition to that, they also have quite limited biogeochemistry. So what that means is that while we can simulate things like surface temperature, there are lots of other environmental factors that we expect to have been very important for marine life in the past, such as oxygen, pH, food supply, and sulfide stress, and all sorts of other things, that a complex global circulation model just isn't going to be able to simulate. And most of the sort of IPCC grade models don't. So that's sort of a challenge that we've been trying to confront. So this is a new model framework that we've been calling Phanero 3D, so Phanerozoic oceans in 3 dimensions, that I've been working on with Ben Mills at Leeds and Erin Saup at Oxford, where essentially we've been developing a new coupled model framework that allows us to not just simulate 3D marine temperature, but also things like 3D marine pH, dissolved O2, that kind of thing. And the way we do this is basically, as many new models are, combining a number of excellent existing models. So taking circulation fields from those more complex models that were ticking over on our screen a second ago, we're able to feed in those physical conditions, so how winds are behaving in response to things like CO2 and topography itself, bathymetry, and ice sheet extent. From other longer term models, we're able to feed in key important things such as boundary conditions with respect to atmospheric oxygen, nutrient supply, and marine sulfate. And then using CGenie, my favorite model, the carbon-centric grid-enabled integrated Earth system model, we're able to then, using the things that we gain from those models, simulate things like the 3D distributions of marine temperature, dissolved O2, pH, organic carbon. Today, I'm going to spend a little bit talking about how we do this, and then a bit more talking about how we might actually manipulate the outputs of this sort of thing and how we might make them most useful to paleontologists, but paleoscientists more broadly as well. So as a teaser of where we can get to, this is, as I point out in my title here, a nearly finished product. So we're able to start simulating a full reconstructive stage-by-stage series of coupled 3D ocean temperature and dissolved oxygen fields. So here, okay, sorry, my globes do spin. Here we have modeled equatorial surface temperature through the Phanerozoic, and then we have modeled dissolved oxygen in shelf environments through the Phanerozoic as well. I will explain how we get there. And then we're also able to do things like start directly linking fossil occurrences to these simulations of dissolved oxygen or whatever other variable you're interested in through Phanerozoic time. Obviously, what we get out is only going to be as good as we get as what we put in. So these are experimental simulations, but we are able to start getting a lot more resolution, particularly in terms of heterogeneity between different environments and different parts of the Earth through Earth history. So this is sort of where we can get to, but let's talk a little bit about the methods. I wanted to dig a little bit more into the software and the sort of day-to-day logistics of doing this kind of research, because I think that's the really cool thing, or one of the many cool things about this seminar series. So, as I say, there is a lot of work before that point. So in terms of this new model framework, I laid it out like this, as really being the coupling of these three models, HADCM3, Scion, and CGenie. As I was explaining that, you might have sort of caught on to the fact that what I'm really doing here is taking simulated outputs from HADCM3, from the Scion model, and feeding them into the CGenie Earth system model. I'll flag now, just in case anyone's wondering, a nice thing about these models, or at least the versions of the models that we're using, is that they are actually internally consistent. So they're all built on the same paleogeographies, which helps us feel better about the way that we essentially nest this multi-model hierarchy together. The way we actually make these models interface is firstly dependent on an awful lot of people. So here I've highlighted some of them. So all of the HADCM3 models that I'm using in this study were run by Dan Lunt at Bristol, and the Scion model framework, the Spatial Continuous Integration Evolution model, was developed by Ben Ross at Leeds. And then in terms of CGenie things, there's a whole community of people running the CGenie model that we're dependent on for model development. But Andy Ridgewell is the primary developer of CGenie, and certainly in this process, Fanny Montiero and Pamela Bullock have also been super helpful in helping to develop these pipelines. On top of the people, there's the software, right? And I guess the key thing I'll flag here is that clearly, or it'll be at least clear to some people, that I can't delve into the code that underpins every single, or the software that underpins every single aspect of this kind of modeling approach. So HADCM3 is, as with pretty much all CMIP grade models, written in Fortran. And the reason it's written in Fortran, which is an incredibly old coding language, I looked up today, it was developed by IBM and first documented in 1956. Fortran is the original language. It's incredibly fast as a scientific programming language, and climate models are very computationally intensive to run. So we stick with the fastest languages. Similar approach applies to the 3D modeling that happens in the CGenie system model. On top of that, though, we have a lot of more interactive and easier to understand coding languages that perhaps people will be more used to running or interacting with. So the Scion model is written entirely in MATLAB, for better or worse. And Ben's able to do that essentially because it's a statistical climate emulator coupled to a long-term carbon cycle model, sort of similar to COPS and GeoCarb, if you've heard of those. So the Scion model actually runs with a lot of iteration in sort of minutes, whereas something like HADCM3 may take a couple of weeks on a lot of cores on a high-performance computing facility. For what it's worth, Genie sits sort of somewhere in the middle. It probably takes a couple of days to run a model for about 10,000, 20,000 years. We then need offline pipelines that essentially translate these outputs into something we can use. And that happens either through MATLAB using functions developed by Andy Ridgewell, or in the case of translating Scion outputs and some other things, using R pipelines that I've developed, some of which we'll talk about in a second. So how do we actually get to those models? So CGenie, I guess the first thing to flag is that, at least in theory, anyone can run these models, or anyone can run CGenie at least, or any of these models. All of these models are open source. So CGenie is publicly available. The Genie family of Earth system models is now essentially run and moderated by a community, still principally led by Andy, and is openly available online. In terms of what the model itself looks like, unless you're developing the model, you actually don't tend to interact with this kind of material an awful lot. But what you'll find is that there are many scripts involved in each module of the model. So this is the biogem module, which is the biogeochemistry module. And each of many scripts within that will be thousands of lines of Fortran code. Thankfully, to run the model, you actually don't need to interact with that code unless you're really interested in development. So model experiments are designed and implemented mainly via text files. So most experiments are forced using two different scripts, one of which is a base configuration that tends to tell the model which physical conditions you're going to use. So things like solar constants, bathymetry, wind fields, that kind of thing, which modules of the model you want turned on. And then, and that might be a more general setup of the model that you use multiple times. And then this script on the right, again, only 100 or 200 lines long, is a user configuration. So that's bespoke to a specific experiment, and that's where you might change things like biological productivity schemes, CO2 forcings, that kind of thing. So in terms of actually running the model, while it is written in a language that lots of people don't learn anymore, actually running the model is much more straightforward than some people might think. Once you've successfully run the model, either on a Unix desktop or on an institutional computing facility generally, your results are then output, again, in quite inconvenient files that lots of people don't get training in during their undergraduate. So generally, client model data will come out as netcdf files, which have a lot of problems or a lot of complications in terms of a large array of metadata and lots of different component data sets that have varying dimensionality. So you might have 3D data fields for things like temperature that might vary with depth or elevation, as well as longitude and latitude. There'll also be a time component. The same thing will be true for a bunch of other variables. There might be other variables that don't have a depth or elevation component. They're only sort of in 2 or 3D. And then there are other things sat in these files that are just things like how long was the model run, or what is your depth vector, that kind of thing. So they're piling a load of information into this condensed file format. And the thing is, one of the key things in terms of working with these kinds of models is that actually, you know, as I say, lots of people don't get training in working these kinds of models. And then that means that while lots of people might be interested in working with climate data, it might not be obvious how to use it. So one of the most popular ways to manipulate climate models is using this Panoply software that NASA makes freely available. And it's great, right? You go to a conference like Ocean Sciences or AGU, and you'll see loads of climate modelers that all have similarly stylized plots and are clearly made with this software. And it's great basically for making maps, and particularly if you only want to make a few maps. But for applications like kinds I'm really interested in, it has some limited functionality, basically. So it's difficult to customize. So often if you want to do specific things in deep time, you can come up against the fact that it's really mainly geared towards climate modelers working in the modern. There's no sort of command line option. So it's very difficult to automate, which is both time consuming and annoying, but it also makes you more prone to human error. And then there's no functionality for doing things like matching point data or extracting subsections of the model, which I think is really key for the kind of approaches that lots of paleontologists, geochemists, etc. would be interested in. And the GUI is just a little bit fiddly sometimes. And it requires an annoying Java 11 installation that sometimes can take quite a while to get right. So that's sort of what motivated me to develop this package, the PaleoClimR package, which, as I say here, is a new R package for the visualization, manipulation, and analysis of paleoclimate and paleoceanographic models. So we're currently in a sort of beta testing and documentation phase, with documentation probably being being the key thing and making lots of different functions that were written for various different reasons kind of all exist within the same ecosystem. In my dream world, it would have been ready for today, but I think probably early summer is our hopeful timeline for that. So it is available on GitHub. Definitely user beware right now, but it's available on GitHub. I'm using it all the time, so there is that. And documentation is progressing. If anyone wants the up-to-date documentation, and really can't wait 2 months, please do feel free to email me. So in terms of what we've ended up doing to develop this R package, I'll talk you through it. So this is the climate model framework that I've been working on. And I kind of wanted to walk through some examples of how I think this package can be useful as I kind of talk to you a little bit about some of the science associated with that. So a key thing in trying to generate this reconstructive 3D model series is that the CGenie model is really good at simulating biogeochemistry. So things like dissolved oxygen, organic carbon, pH, wicked. Has a terrible atmosphere. It essentially relies on a 2D energy mass balance model. So we need to take some of the more advanced circulation patterns from a more complex model like HADSI-M3. And that's basically the role that HADSI-M3 plays in this stuff. And we want to know that we've done a good job of representing that circulation. And that's really what this GIF is trying to show here. The key thing here, the topography matches relatively well. I've coarsened it up because CGenie is a coarser model. And then we want to see if the circulation is doing the right thing. It doesn't really matter exactly what the barotropic stream function is. Critically, what we're seeing here is in the deeper reds and deeper blues here, we have either counterclockwise or clockwise surface ocean circulation. And what we're really interested in is how well do they match? And hopefully you can see they match pretty well. There are some actually in the Cenozoic that you might have caught don't match quite so well. We've already fixed that, but I didn't make a new GIF today. But you need to be able to make lots and lots and lots of these plots to do this kind of work. And as you might imagine, this is not the first version we've made of this. So many times you have to create a huge number of plots. And it would take so much time doing that in something like the Panoply software. So this sort of leads me to the first bit of functionality in this PaleoClimR package, and that's just to make maps basically, and to make hopefully publishable quality maps. So this is a function called CGenie map that sits in this is where it sits in the documentation right now. And what it allows you to do is a bunch of things. Well, it allows you to do one thing really, which is plot maps. But it allows you to change a bunch of parameters, and then you can imagine that if you've set these once, you could loop through and do this tens or maybe hundreds of times if you needed to. How this actually looks to run, you need very little information. Really, it's sort of a line of code. You might choose to set a few variables if you're feeling picky before you go into running that one function. But this one function, CGenie map, will then create you what hopefully is a nice publishable quality map, or certainly a map of the quality that I tend to publish. Without this, you'd actually have to do a lot of work. So that Panoply package, that Panoply software is masking a huge amount of computation that's done behind the scenes. So you'd want to set up default variables so you're not, between ocean temperature and ocean O2, you're often jumping 6 orders of magnitude between Celsius and micromoles per kilogram. So that can become very annoying and fiddly. Results are often stored in huge nested directories of model results, so you have to actually track them down. Unpacking netcdf files can be a real pain and takes a bit of getting used to with packages like rnetcdf. And then you actually have to start constructing data for mapping. So if you want to plot something like this in R, you want to translate 3D arrays that you're extracting alongside lots of metadata information into data frames that R can handle well. You want to deal with boundary conditions. And then a key computationally intensive thing is you need to create an awful lot of spatial polygons. So if you want to actually be able to project this data in a bunch of different map projections, That's going to take a lot of manipulation by packages such as SP and SF. Essentially, what we need to do is loop through all of the different cells in a global climate model, which would be thousands, if not tens of thousands, and assign each of those a polygon within this SP and SF network. Then you might also need to apply a reasonable amount of logic to start identifying what is land, what is ocean. If you're interested in specific environments like shelf environments, that's going to raise flags that I'll talk about later. And then you need to go through a lot of projections. So often this is tens of lines of ggplot2 code as well. So there's an awful lot happening within what becomes just one line of code in this mapping thing. So we could add variables to this, but ideally the idea behind this framework is essentially it isolates the variables that people would be most interested in manipulating so that they can then produce lots. We can do the same thing for HATCM3. So here's the HATCM3 map function with some added functionality to plot topography, top of things, because HATCM3 is really nicely resolved. And then I'm working on versions for SCTs DEM, CSM versions. I'm sure I'll do the same for other paleotopography projects that I know are currently in progress. We can also deal with difficult things about climate models that are just quirks of climate models, really. So often if you have a surface parameter in your climate model, it will completely leave out your continent, your land-ocean mask. And if we're interested in things like that barotropic stream function, that circulation metric, we probably want to know where our landmass is. So instead of plotting it up in something like the NASA software, with these custom functions, you can then overlay your landmass. So if you're interested in surface air temperature or stream functions, it becomes much easier to interrogate your model results. The next step to actually producing a 3D series would be to force the model with best guess reconstructions at each point in time. So we're doing this by stage, which means essentially we have to draw a lot of different points along these curves and extract data and extract estimates that we would then use to force the models. So that's what goes into those two sort of 100 or 200 line text files that I was flagging earlier on. And I guess what I would flag is that actually, if you're doing this all the time, if PaleoClimate modeling becomes your bread and butter, automating this process is something that's really useful and something that I've developed functions for that will be included in a later release. The other thing you have to do is when you're running these experiments, you have to ensure that they reach steady state. So you have to ensure that actually you're representing a stable version of the Earth system. When you initiate a climate model, it starts as we say, from cold. So you start with a sort of fully uniform global environment, generally, a fully uniform cold ocean, and you need that ocean to start circulating and then reaching a stage where things like global marine O2 have reached steady state. Often, this means looking at hundreds, if not thousands, of plots like this to ensure that you're designing functionality in the package that allows us to do things like that as well. And then it sort of gets to the fun bit. I just got where we can start extracting information about what's happened through time. So here I'm showing equatorial sea surface temperature from the models as compared to these climate bands representing the SCTs, Mills, and Donahue series from, what was it, Science Reviews, from 2021. And what we find is that actually our simulated surface temperature is a very good match for that. You don't need to worry about the absolute magnitude right now. That's impacted by the grid distribution. But we want to be able to generate plots like this to see how well we're fitting our proxy records. And again, to do that means delving into these really big files and extracting key bits of information. So that's where functions like this CGNE data function become really useful. We can go in, extract what the surface temperature is for every single model in a neat little loop. And in no time, you've gone through and extracted equatorial sea surface temperature for all 105 models in this series. Without this kind of package, that would take a huge amount of work and would be very difficult to replicate. I'm conscious that I'm running a bit low on time, but definitely worth speaking about ocean oxygenation, how we start thinking about that. A lot of my research is motivated by understanding how dissolved O2 in the oceans has changed through time, and the history of deoxygenation and oxygenation in the oceans. And really, in the sort of first study related to this model series, I'm trying to get at this problem where most marine animals live in very shallow environments, and most geochemical proxies are recording that sort of deeper, the sensitivity of these proxies is really focused on these low O2 shelf slope settings. So again, using this new package, we can start delving into these different sort of spatial locations in the model. So what that means is that we can go in, cut out all of the shelf cells in our model, so around the continental boundaries, and then see how many of those shelf cells are anoxic or very low O2. We can then also look at what dissolved oxygen concentrations are like in non-OMZ shelf environments, so the ones we expect most marine ecosystems to be existing. We can also start computing metrics that we might reconstruct from the geochemical record to allow us to compare our simulated models to our geochemical records. And I'm just going to switch slide here. But here I've got the extent of anoxic seafloor on the top plot and the extent of suboxic seafloor on the bottom plot. And what we see is very similar to what we've reconstructed using trace metal records. They're fully independent of this, where essentially in the mid-Paleozoic, we get this huge reduction in the extent of anoxic and suboxic seafloor, which essentially amounts to a huge oxygenation of the seafloor. So for the first time, we're able to use just physics and our long-term carbon cycle models to produce a really quite good match for the shift that we observed based upon trace metals on organic carbon records, which we've been really pleased about. But again, we have to actually compute these metrics. And using the sort of traditional framework of going into graphical user interfaces and interfacing with these models that way, that would be incredibly difficult. So again, we need to write functions that do these things. So we need to go into the model and drill down in each long lat cell and say, what is the dissolved O2 concentration in the bottom waters of this geographic location? And how does that fit within the redox frameworks that geochemists often use? So here, that's exactly what I do in this CGenie bottom water redox function, is go through, cut out all the seafloor and assign cells to either anoxic, suboxic, or oxic conditions. You could vary those thresholds, but this is the kind of thing that people like me and Alex Pohl have been doing all the time, and now it's wrapped up in a nice little function. And I guess the sort of final thing to emphasize here is that actually dealing with shelf environments can be really difficult. I think this is really important because the locations where marine animals can live is obviously dictated by the positions of the continents through time. And as the continents have moved through time, that also impacts, sort of how much equatorial and tropical ocean conditions are experienced by different marine animal groups versus more polar conditions. And trying to actually model these shelf environments has historically been a bit difficult. When we published this paper in PNAS a few years ago, I actually went through each continental configuration we had and, for my sins, cut out the shelf by hand. I went through every single cell and asked, are you directly next to land and within the top 3 layers of the ocean model? And if it was a yes, it went into a very long vector that we then isolated in an R function for each one. And these bespoke masks took ages to make. Now we have a really cool function that I partly wrote, but was also my my student Prem Griska did an awful lot of the work identifying the coastlines. Now we can do this for absolutely any continental configuration in a matter of seconds. So this CGNE shell function is awesome for specifically trying to isolate the environments we expect marine animals to have been living. And I guess the sort of final point here would be, we're now at the stage where we can also do this with point data and start saying, okay, you know, historically, what I've done in a lot of my studies is isolate these shelf environments. And I think that's really useful, and it's an interesting end member perspective because our fossil record is unevenly sampled. And sometimes we may have had animals living in a particular location and we just don't have a record. So I think that's one great end member. The other end member is, where do we have fossils and what were they experiencing? And we need functions that can do that too. And I guess the sort of, we're now working on it doing exactly that and comparing those end members that I just introduced actually across the end Permian mass extinction, where we see a lot of geographic bias in where our, or geographic unevenness in where our fossil record comes from, particularly where we have continuous sections. So working on functions in the package that can do both point matching to climatologies, but also do that thing where we go around and cut out all the shelf conditions so that people can compare what their simulated model means for the shelf environments that we think existed versus what it means for the animals that were, or organisms we're actually seeing in our sampled fossil record. We can do a bunch of stuff to summarize that, but I think I'm over time, so I'm going to try and finish there. Happy to take any questions. Again, if you can, hold on for a couple of months and this will be available. If anyone is keen to use it straight away, drop me an email. I'm happy to chat. Finally, I just want to thank all these people, and particularly flag, you know, I think in a seminar about software things, it's important to flag that so often it's not just your co-authors that are really important, it's the people that you spend time coding with or next to or around, who you can ask difficult questions to, turn to when you're stuck. And all of these people and more have been very critical in my ability to do that. So very grateful for them. Right, I'll stop talking now.
Brilliant. Thank you so much.